Chapter 2, Sexual Reproduction in Flowering Plants. Summary. Flowers are the seat of sexual reproduction in angiosperms. In the flower, androecium consists of stamens, represents the male reproductive organs, and gynoecium, consisting of pistils, represents the female reproductive organs. A typical anther is bilobed, diathecous, and tetrasporangiate. Pollen grains develop inside the microsporangia. Four wall layers, the epidermis, endothecium, middle layers, and the tapetum, surround the microsporangium. Cells of the sporogenous tissue lying in the center of the microsporangium undergo meiosis, which is called microsporogenesis, to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represents the male gametophytic generation. The pollen grains have a two-layered wall, the outer exine and inner intine. The exine is made up of sporopollenin and has germ pores. Pollen grains may have two cells, that is a vegetative cell and generative cell, or three cells, that is a vegetative cell and two male gametes at the time of shedding. The pistil has three parts, the stigma, style and the ovary are present in the ovary. The ovules have a stalk called a funicle, protective integuments and an opening called micropyle. The central tissue is the ocellus in which the archisporium differentiates. A cell of the archisporium, that is the megaspore mother cell, divides meiotically and one of the megaspores forms the embryo sac, which is the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate. At the micropylar end is the egg apparatus, consisting of two synergids and an egg cell. At the chelazal end are three antipodals. At the center is a large central cell with two polar nuclei.
Pollination is a mechanism to transfer pollen grains
from the anther to the stigma. Pollinating agents are either abiotic, for example wind and water, or biotic, that means animals. Pollen-pistil interaction involves all the events from the landing of pollen grain on the stigma until the pollen tube enters the embryo sac. That is when the pollen is compatible. Or pollen inhibition means when the pollen is incompatible. Following compatible pollination,
the pollen grain germinates on the stigma and the resulting pollen tube grows through the style, enter the ovules and finally discharges two male gametes in one of the synergids. Angiosperms exhibit double fertilization because two fusion events occur in each embryo sac, syngamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus. In the primary endosperm cell, zygote develops into the embryo and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes the development of the embryo. The developing embryo passes through different stages such as the proembryo, globular and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomics have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony. Thank you.